So I'd like to welcome everyone to the session. My name is Greg Downey. I'm a pulmonary critical care physician based at National Jewish Health in the University of Colorado in Denver. I'm the director of the LAM clinic in Denver, and I also have the honor of being the chair of the LAM Foundation board. But I'm not here in that capacity today. I'm here in a capacity to introduce probably the leading light in the LAM community, Dr. Frank McCormick, who is a friend and colleague. I first met Frank when we were training together in the laboratories at National Jewish Health when we were doing our pulmonary fellowship. And it was apparent to me even then that he was an incredible leader. His project was one looking at some very basic aspects of cell biology in a very technically difficult area, but he excelled at it. He left to go to Cincinnati, I left to go to the University of Toronto, but our paths would cross again on a Friday afternoon when I got a phone call saying, would I like to join a group of people who were trying to figure out this disease called lymphangioliomyomatosis, and he and Sue Burns were forming the LAM Foundation. And I would say that for 25 years, Frank was the scientific director of the LAM Foundation and moved the field by legions. He is an international leader, not only in pulmonary critical care medicine and rare lung diseases, but in LAM. His publication record is stellar. He has over 200 publications in the highest profile journals, and he has a long list of awards, including the American Society of Clinical Investigation, the American Association of Physicians, the ATS Distinguished Achievement Award, and the Alpha Omega Alpha Award. As I mentioned, he was the scientific director of the LAM Foundation for 25 years, and in that time, he had the ability to bring together basic science and clinical science to further the field of LAM treatment. And he was the principal investigator and the force behind the MILES trial, which showed the efficacy of sirolimus in the treatment of LAM. I won't belabor the points in Frank's accomplishments, but suffice it to say that he is really the leading light in the field of LAM, and today he's going to talk to us about the future of LAM. So without further ado, I will welcome Frank to the stage.
Well, I can't thank Greg enough for that introduction. I don't think I've ever had an introduction like that. I certainly appreciate that and also appreciate the invitation to speak to you today about the future of LAM. And in particular, I'd like to thank the organizers, and I know I'm going to leave somebody out, but Kayla, Simon, Lisa, Pat, Patty, Greg, Vera, Harry, and many others on this list. It was very kind of you to ask me to speak. This meeting's always been very special to me. So many people that I've met on the phone and as patients in clinic turn up at this meeting, and we become friends and see each other year after year. When I started as scientific director, Holly and I had 5 children, including a 1-year-old. And when I stepped down as scientific director, we had 5 grandchildren, including a 1-year-old. And working on LAM has left me with a sense of joy and fulfillment that money can't buy, and I thank all of you for that. LAM is a family, and families are the engine behind the LAM effort. When Pat Gentile called to ask me to speak, she recommended that I make this an uplifting talk and family-friendly without a lot of scientific jargon, which I'll do. And I'm, of course, going to include a few lame jokes and irrelevant pictures. So I can look down there. So these are my disclosures. I can't see that far, actually. I used to be able to. When I started, I could see that far. I have a patent for the use of VEGF-D. Lisa Young and I have a patent for the use of VEGF-D as a diagnostic test, and all royalties have been waived to our institutions. We have a patent pending for hydroxyapatite fluoridesis and grants from various agencies. And a few drug companies provided the drug for our trials. And I was scientific director for 25 years of the LAM Foundation. This is a talk about the future, but I have just 2 slides about the past. I wanted to review some of the progress that's occurred in the last 30 years, especially those that were LAM Foundation inspired, supported, or initiated advances. So everyone knows the beginning of this story. The foundation was founded by Sue Burns and her husband Fran in 1995. They lobbied the NIH for an NIH registry and an intramural program, which continues today. We developed a peer-reviewed research program, and Sue organized the patients in many ways, including the LAM Liaison Network. The CAST trial, which tested sirolimus in patients with kidney tumors, was supported by the LAM Foundation and also showed a promising signal in the lung. The LAM Clinic Network was formed. Lisa Young and I have developed VEGF-D as a diagnostic test that enabled us to fully enroll the MILES trial, which demonstrated that sirolimus was effective for treatment of LAM, and FDA approved the drug in 2015. The ATS and ERS LAM guidelines were written by many people in this room, and single-cell transcriptomics have really transformed the field from several groups in Boston and Cincinnati and elsewhere. Sirolimus is now approved in over 40 countries and counting. The MILE trial is just completing, and I was hoping to be able to tell you the results, detailed results, but they're just not ready, so I won't be able to do that today. And of course, here we are at the 30th anniversary. This remarkable progress is a result of a coordinated effort by all of us, patients, families, clinicians, investigators, and trialists, and the 30th anniversary of the Foundation provides the opportunity to reflect on all that has been accomplished. These are the accumulated assets as I see them today. We have a highly motivated, well-informed patient community, highly effective patient advocacy organizations in the United States, in the UK, around Europe and Asia, a skilled, engaged, purpose-driven research community looking for answers for patients, a global network of expert centers for clinical care and research, peer-reviewed programs for pilot funding that leads to more substantial funding and ultimately to clinical breakthroughs, a solid understanding of the natural history of the molecular basis of LAM, LAM clinical guidelines from multiple societies, clinically useful biomarker, and an effective suppressive therapy. This is a very powerful tool chest that provides a wonderful foundation for the next 30 years. And we have these 4 secret weapons, which have been true from the beginning, that make me so optimistic about the future. LAM is a very motivating diagnosis. It catches all off guard, surprises, shocks, stuns. It affects our mothers, sisters, and daughters in the prime of their life, with family and careers at full tilt. And that demographic is so powerful because the disease moves slowly and there's time for all to engage, and 3 generations are often involved in the fight. Our collective inspiration to get patients and scientists together at this meeting has been transformative. And the LAM cellular and molecular pathway is so central to biology that the world's best scientists study it, and that's just good luck. So we've got to get back to the future. That's what this talk's about. So this is my idea for a new LAM patient in a clinic visit in the future. The doctor says, how fortunate we caught this early enough through screening. We have 3 proven effective therapies to offer you that can arrest disease progression and hopefully induce remission. Your panel of markers suggests that you're most likely to respond to treatment A. We can do some blood tests in a few months to determine if treatment A is working for you as planned. If not, or if treatment A bothers you, we may try treatment B or C. If we run into trouble, we may have to do additional tests on blood or tissue to identify the unique Achilles heel for your particular LAM cells. Luckily, we have targeted therapies for many of these. I believe these are achievable things in the next 30 years. And I've listed them as our tangible goals here, divided our future goals into two categories: the most tangible goals and the more aspirational goals. For tangible goals, I've included improved early detection and intervention, better tools for diagnosis and disease tracking, a more thorough understanding of disease pathogenesis, improved trial design, access and feasibility, optimized disease management and quality of life, remission-inducing therapies, and personalized approaches to care. These are the more aspirational goals. They include gene therapy, stem cell therapy, lung regeneration, tissue engineering, or xenotransplantation, that is taking organs from other species, bioengineered scaffolds. These are, I think, in the fairly distant future. It's a little hard to see which of these might emerge as a therapy, and I debated whether to put them in at all because I worried about generating false hope or some kind of hype, and I certainly don't want to be viewed as this kind of a scientist. So in terms of improved early detection, we need to protocolize LAM screening in all TSC clinics. There are efforts afoot to do this now, but they're not as organized as they might be. We need to raise LAM awareness in nephrology, urology, and OB-GYN clinics and communities. When patients are seen in our clinics with an angiomyolipoma that was recognized 20 years earlier, but they had not received a diagnosis of LAM and no one thought of it then, that's a fairly common occurrence in our clinics. Patients who have repeated pneumothoraces often go undiagnosed for long periods of time. And we need to get the word out that people should be thinking about LAM in these situations. We need to capitalize on incidental lung cyst discovery, on CT abdomen and chest. And this could be done now. We could be doing AI screening of CT reports that mention lung cysts or angiomyolipomas. That can be easily implemented in hospitals around the country to raise the issue of LAM in cases where the CT may have suggested it and no one thought of it. And it's also possible to use screening techniques that employ machine learning to scan CT scans, just CTs, to suggest to physicians caring for patients that their CT may indeed represent a disease they've never heard of, this lymphangioliomyomatosis. We should be ordering chest CT for young women who present with a first spontaneous pneumothorax. At least we believe that to be true based on a cost-effectiveness study we did some time ago. And certainly all women who present with recurrent pneumothorax or that history of recurrent pneumothorax identified by AI screening of an electronic health record should be screened. We need to develop better tools for diagnosis and disease tracking. I think our most important single development would be to find a way to measure total LAM cell burden. That came up several times in our scientific session today. We need to find a way to identify LAM cells by PET so we can find out if our treatment is killing or disabling LAM cells in the body. And PET and MRI are among the leading candidates for that role. We need accessible automated methods to quantify lung cyst burden. Those are also possible today, and could be implemented in our clinics and in our hospitals. Currently, there's expense and other issues involved. We need accurate, user-friendly tools to track lung function trajectory at home, including spirometry, oximetry, actigraphy, step testing, and other home monitoring techniques that can be shared with doctors. We need improved LAM-specific blood tests for diagnosis and prediction of disease progression and treatment response. One way that this is being approached in Cincinnati is that Minzy Guo and Yan Zhu are looking at multi-omic analysis where they're integrating data from blood protein markers in LAM patients with gene expression in the lung and the DNA regions that are activated to identify the most promising markers to validate in our samples for LAM diagnosis, progression, and response. This is a sophisticated way to combine all kinds of data in an integrated way so that we can pick the most promising candidates to test in our studies. And much of these data are being made publicly available through the LAM Cell Atlas, which is an incredible tool that was built by Jan and Minzy that is available to anyone, and very few rare disease communities of this size have this kind of a resource available to them. We need a more thorough understanding of disease pathogenesis. We need to characterize destructive processes in LAM to identify critical vulnerabilities. We need to find ways to target the feedback loops and escape pathways that limit effectiveness of our therapies. We need to define the role of immune surveillance in LAM cell clearance. We need to determine the role of sex hormones in disease pathogenesis. And we need to identify and confirm the source of LAM cells. Understanding these elements would help us to design better therapies and move the field forward. We need better trial designs. The MILES trial took 7 years, cost $8.5 million. We had many delays involved with it. There has to be a better way. We need to find a way to, we need to distinguish between LAM cell burden and LAM cell activity. Lung dysfunction in LAM is due to both a reversible and an irreversible component, a reversible component being cellular infiltration, and the irreversible component being the cystic remodeling that has already occurred and which may not be repairable. So we have to understand that we're looking for induction of remission, not necessarily a complete cure that could restore lung function, at least not in 2025. Perhaps sometime in the distant future with some of these more aspirational goals. Reducing barriers to conducting trials, centralizing the IRB, doing harmonized international trials, accelerating approval pathways, developing surrogate endpoints, using home monitoring and patient-reported outcomes, and developing trial networks like the Cystic Fibrosis Foundation has. That has greatly accelerated drug discovery in that group and really transformed the field. Cystic fibrosis is a completely different disease than it was when we were training. We hardly ever admit patients to the hospital anymore with cystic fibrosis. It's just been amazing. And it's been due in part to their ability to do trials in a rapid way. And we need to move beyond underpowered trials with safe, well-tolerated agents. All of us have done trials with very safe drugs that are easy to get funded and that end up in a space where they're not taken forward and don't contribute to therapy. And I think we need to focus on the most promising agents and design trials that have the potential to actually change patient outcomes. These are the LAM trials that have occurred to date. You can see here that there have been a number of them. And really, most of them haven't resulted in changes in therapy. We don't use most of these agents in the therapy of LAM, really none of them, other than sirolimus at this point in time. We need better endpoints. Aspirational endpoint, again, would be this LAM cell burden by PET or MRI. That would greatly accelerate our trials, enable us to do combination therapies with sirolimus and other trial designs that could find answers for patients with LAM. The most plausible available endpoints include FEV1 and change in tumor size, angiomyolipomas and lymphangiomyoma size. These are really surrogate endpoints. They don't really address how patients feel, function, or survive, which is what the FDA is looking for, but they're the best we have to date. There are endpoints with limitations that we consider in many trials. Many of them are used as secondary endpoints, DLCO, VEGF-D, or other biomarkers, patient-reported outcomes. And then some endpoints are not really plausible with LAM because survival is so long that survival as an endpoint really is not statistically possible. And a 6-minute walk test is useful in LAM care, but its test performance is really not adequate to use for a trial endpoint, as it has been, say, for pulmonary hypertension. We do have candidates for trials that are not on sirolimus. In that group, it'd be possible to do more randomized controlled trials, and they include patients with early mild disease, patients who've just decided they don't want to take sirolimus, have read about it and don't like the side effect profile that they've read about, patients who've been intolerant to sirolimus, and patients who are resistant to sirolimus. So there are patients who are available for a randomized trial that compares a placebo to a novel agent. The sirolimus otherwise complicates randomized controlled trials because we'd have to do combination therapies in patients who are already on that drug, and it's going to be hard to see a signal in patients who are well stabilized by sirolimus alone. Sirolimus, of course, is not the ideal therapy. It doesn't induce remission. It doesn't kill LAM cells. It simply suppresses LAM cell activity. What we really need is an agent that kills LAM cells. And we need to think about novel rare disease trial designs, including using historical controls, concurrent controls, including N-of-1 trials, adaptive trials that change in protocol during the course of the trial as you learn more about how patients should be randomized, what numbers you need in each group. You can change the design as the trial goes forward. Basket trials where we're studying, say, all patients who have mTOR mutations in various diseases, including tuberous sclerosis or LAM or Dipnick or sarcoid or other diseases where mTOR is activated, factorial designs and crossover designs. These are all available to reduce the number of patients that would be needed to answer a question. In terms of optimizing therapy and management of disease and quality of life, we're working on refining mTOR therapy now through the MIDAS and MILD trial. And again, I was hoping to tell you more about each of these today, but it's going to take a little more time to actually make a full analysis of the data. But our hope is that we can define the side effect profile through time, through MIDAS, determine the efficacy over the course of many years, and through MILD, determine if we should be starting the drug earlier to suppress disease progression before severe damage occurs. We're trying to refine our approach to pregnancy, including uses of IVF and surrogacy. There's a LAM Foundation Women's Health Task Force led by Steve Hammes, who's looking into this. Determine the safety of mTOR inhibitors in pregnancy by establishing a registry. We're accumulating some experience with the use of sirolimus in pregnancy, and it's really probably the only way to know if this drug can be used safely through time is by collecting data from patients who decide to take that course. We need a better pleurodesis agent than talc, and I'll talk a little bit about that in a moment. And we need to update the LAM guidelines. I think possible topics, and we may not have accumulated data for all of these now, but maybe going into the future, we'll be able to recommend earlier treatment with sirolimus, maybe make pregnancy recommendations. Looking at recurrent pneumothorax as an indication for sirolimus. That's not something that we've routinely employed, but there is evidence to suggest that sirolimus suppresses the incidence of pneumothorax. Earlier mental health intervention, and this is something that I feel strongly about. I've watched hundreds of LAM patients have a very bad year after they're first diagnosed with LAM. It's a very predictable pattern, and that the return to joy can take 1 or 2 years. Why do we just wait for that to happen? Why do we watch it happen? Why don't we proactively intervene? I've never really... And a little more information about management of Achilles effusion might be needed before we can make specific recommendations about lymphatic imaging, but it's become very useful in many LAM patients to understand exactly where these leaks are coming from and how they could be blocked through embolization and to design other therapies to try to deal with refractory chylothorax. And lymphatic imaging, especially at the University of Pennsylvania, has really been useful for many of our patients. So the MIDAS trial has enrolled over 400 patients. I appreciate everyone's participation in that study. We've collected almost 1,000 samples. Our hypothesis is that sustained treatment of LAM with mTOR inhibitors is safe and durably effective. It's obviously a real-world observational study. We're not telling clinicians what to do, we're just watching what clinicians do. In preliminary look at the data, there is a difference between groups in that the patients who are on sirolimus appear to be stabilized. There are many possible biases and confounders here, and we're working through the data to understand those before we present this in a formal way. These are preliminary data, and we've been showing them for a few years now, but soon we'll have the actual data that's been fully analyzed available for everyone. And then the MILE trial was an 8-year-long trial that looked at early low-dose sirolimus in patients with LAM who had normal lung function to determine if we can arrest progression in disease before more advanced stages occur. Patients are randomized to 1 milligram of sirolimus or placebo, and they're followed for 2 years with serial lung function every 2 months. There are many sites that have participated in this study. We were able to reach our targeted enrollment despite conducting this trial through COVID, and I give great credit to Susan McMahon and Carrie Geininger, who kept things going through that very difficult time, and we're hoping to report the results this fall. Now, with respect to a better particle for pleurodesis, we've been testing particles that are basically little tiny spheres of bone that are just a few, say, 25 microns to 45 microns in size, very tiny, that are placed into the pleural space and induce pleurodesis in the same way as talc, but they're biodegraded in the same way as your body degrades bone as it's remodeling after a fracture or when you go for a run. Your osteoclasts are degrading bone and your osteoblasts are building bone. Well, here osteoclasts clear this particle over just a few months after it's placed in the chest. And without going into detail about what this data shows, just suffice it to say that in a mouse, this particle protects from pneumothorax more effectively than talc, and it's removed from the body within about 2 months. So we're hopeful that this could be a better solution for patients with LAM, and that perhaps the bleeding complications that occur in patients who go on to need transplant won't occur as often, and the chronic pain that some people feel after talc pleurodesis won't happen as often. And you won't be exposed to the lead that's contained within these particles, within talc particles. And so I think that we're actually quite excited about this, although my fellows tell me that this is not rocket science, this is rock science. And just to say that it also works in pigs. Now, these are the same particles that are injected for cosmetic purposes to make lips more appealing. And the way they make them non-inflammatory is they heat them to 1,000 degrees. And interestingly, if you don't heat them, they're very inflammatory and they induce pleurodesis in the pleural cavity. If you heat them to 1,000 degrees, they're no longer effective for pleurodesis. So we're looking at different temperatures and durations of heating these particles to find the optimal balance between inducing pleurodesis and having a clearance period that's long enough to induce a durable pleurodesis before they're cleared. Now, perhaps one of the most important goals for the future is to discover remission-inducing therapies. And these include very promising candidates, such as metabolic inhibitors, mesorbine, which is a nucleotide analog that gets incorporated into DNA that kills cells that are actively growing and which has shown promise in the lab. There are many other examples of those kind of nucleoside analogs that have promise. Multiple kinase inhibitors, there's rapamycin analogs that are linked to other drugs together that can inhibit the mTOR pathway more effectively and block some of the parallel pathways that defeat our therapies. Immune-targeted therapies such as checkpoint inhibitors that there were many talks about this today that are really coming to the fore and look very promising for treatment of LAM. Lymphangiogenesis inhibitors like pazopanib, that's a drug that was incidentally given to a patient in LAM in Japan and actually reduced the size of their angiomyolipoma in a case report. That's, I think, a very interesting finding, and it makes biological sense because it inhibits lymphangiogenesis, which is an important part of LAM pathogenesis. Hormonal modulation. We still have not cracked the conundrum of what role sex steroids and estrogens play in LAM progression and pathogenesis, but it makes a lot of sense to test these ideas, the same kinds of ideas that are used in breast cancer in LAM, and then blocking matrix-degrading enzymes with various protease inhibitors like CTSK inhibitors is a promising future direction as well. And developing personalized approaches to therapy and care. We need to develop biomarkers that predict disease progression and treatment response, tailor therapies based on endotypes, or is the patient premenopausal or postmenopausal? Do they have lymphatic involvement or not? Do they have the small cyst phenotype that generates very low DLCOs, or do they have large cysts that affect FEV1 out of proportion to DLCO? Nishant and others have developed risk calculators that can be used to predict disease progression in individual patients. Dose-adjusted therapy can be dose-adjusted based on biomarkers, and this is another idea that's being tested, and an application for a grant has just gone in to test the idea that you might be able to use VEGF-T to decide what the right dose of sirolimus is. There's promise in using metabolic profiling and other pharmacodynamic measures to determine whether the drug's hitting the target, whether we can use a lower dose, whether we need a higher dose. And then examining blood markers and lung tissue of non-responders to identify escape pathways and activating mutations that may need additional thought about inhibitors that could be used to block these pathways that interfere with our ability of our drug to hit the target. In terms of more aspirational goals, starting with gene therapy has been approved for a few bone marrow, muscle, eye, brain, and metabolic conditions where genetic defects are corrected and the cells are returned to the body. It's been tested in mouse models for LAM. For human lung disease, this is something that will likely be worked out for cystic fibrosis and other more common diseases first. And we have some LAM-specific barriers, including that specific targeting of LAM cells may be difficult because they're so close to normal cells. And how can we limit the off-target effects if we can't clearly distinguish a LAM cell from a normal cell? The size of the TSC2 gene is large and makes it difficult to package into vectors to use for gene therapy. Access to LAM cells deep in the lung, nodules, and in other niches around the body may be difficult. And then the same immune responses that defeat gene therapies for every other lung disease apply to LAM as well. Lung regeneration is a concept that we could induce the lung to reactivate programs that build alveoli as occurs during development. And this requires coordinated orchestration of complex programs, cell-cell crosstalk, growth factors and cytokines, modulation of gene and signaling pathways. And stem cell therapy is similar, except that instead of coaxing the patient's own cells to rebuild alveoli, this involves introducing progenitors for alveolar epithelial, endothelial, and mesenchymal cells. And the biggest challenge for these two therapies, for both of them, is that recapitulating this 3D structure of the lung in a mature organ and basically trying to recreate development is a challenging concept. You can see here a branching, conducting airway that ends in these grape-like clusters of alveoli, and how we have millions and millions of them within a whole lung. And they develop in the embryo by a series of budding and branching events that is, I think, going to be challenging to recapitulate. And it's a lot like building a skyscraper. It goes up in a certain order, and it's an orderly progression until the building's complete. It's similar for the lung. So stem cell therapies, alveolar regeneration are promising. There are some hurdles, but it's definitely something that we can hope for in the future. Bioengineered scaffolds. These are whole lungs that are decellularized with detergent. So we take all the cells off of this connective tissue matrix that retains the lung's architecture, and they're recellularized with the patient's epithelial, endothelial, and stromal cells. This is reported to have worked in a rat functioning in gas exchange for 45 to 120 minutes. This was published in Science in 2010. And what they did on the left, I don't have a pointer, but the lung from a rat was removed, washed with detergent to remove all the cells, placed in a chamber where the trachea was hooked up to one circuit and the vascular tree was hooked up to another circuit. Into the trachea went pulmonary epithelium, into the vascular circuit went vascular endothelium. The lung was recellularized, placed, transplanted into a rat who was able to, and there was gas exchange that was present for up to an hour or so. So this is, I think, a fantastic, in the sense of a sort of magical, fantastic idea for a future therapy for lung disease, but it's something that people have been thinking about. And then xenotransplantation, such as transplanting animal lungs into humans. Pigs are a good size match for humans in terms of lung size. Non-human primates transplanted with pig hearts, kidneys, survive for weeks or months. Challenges for the lung are that non-human primates transplanted with pig lungs only survive for hours. And that's due to hyperacute rejection. And something we found in our pig experiments, they're very prone to pulmonary edema. They have very fragile pulmonary capillary networks. And of course, pigs are often infected with a variety of things. So this is another, I think, distant possibility for a therapy in the future that's interesting to think about. So in summing up, what LAM has is hard-won, precious, and delicate. It needs to be cultivated and nurtured and never taken for granted. What makes this LAM effort so effective is full immersion patient engagement. Donating tissue, filling out questionnaires, enrolling in trials, participating in meetings, supporting research are the fuel for this rocket ship of progress. Every caregiver and scientist here will tell you that it's an absolute privilege to work on LAM. And the Foundation's reputation in the pulmonary community is solid gold, and we need to do all we can to preserve it. LAM Foundation funding represents hope, recruits new minds to the field, facilitates federal funding, and forms the basis for trials and clinical advances. And goals for the future are to identify patients earlier, develop methods to quantify LAM cell burden, refine current approaches to therapy and management, and find new remission-inducing therapies. We have tremendous momentum, and the future is bright. Yeah, I don't need to say anything about this. But what I really feel about the last 30 years of what LAM has done for me is that I'm grateful for the people I've met, the daily lessons I've received in courage and resilience, the opportunity I've had to talk about LAM all over the world, all I've learned about science and medicine from LAM, the privilege of reassuring patients every day in clinic, excuse me, to have had such meaningful work for 30 years that LAM has been part of my life, and to have had the support of my family throughout. So this was us at my youngest son's wedding in May of 2024, and he and Kate have blessed us with a new addition. This is Jack. He likes yogurt. And I think he's going to have the same kind of expensive Thai dry cleaning bills that I have. And thanks to everyone. Thank you to all my colleagues, collaborators, and patients, starting with LAM patients, with Sue Burns, the LAM Foundation, Nishant and Jane and Ninzi and Jan, all the colleagues that are present here from the scientific community that we've interacted with so often over the last 30 years, Lisa Young, Miles, Mild, Midas teams, LAM clinic directors, and our funding sources. And thank you for listening to me. Thank you so much. Yeah. That's it.